So I'll be talking on essentially the basics of Onivyde. The data has been out there for a while now, so I'll just cover some of it along with some points on advanced pancreatic cancers. The whole idea is that while the basic molecule is still irinotecan, there is this liposomal component of it which does ensure that, through the pathway there is increased exposure to SN38, which is the active moiety in this drug. So that's the whole idea of nanoliposomal pegylated irinotecan. Some of the PK/PD is also a little different. You have greater persistence in circulation for NAL-IRI. It's almost 1 week as opposed to less than 1 hour for conventional irinotecan, and there is prolonged SN exposure, and that is why the dose needed to achieve intratumoral SN levels is probably lesser when you give Onivyde. That's the basis of the use of this drug. Now the dosing is slightly different if you use it in first line or you use it in second line. It's in first line, when you're giving it, it's essentially 50 milligram per meter square and 70 milligram per meter square. Otherwise dilution can be dextrose based or normal saline. So essentially we end up using 2 to 3 vials per patient per dose. That's how it goes. Now pancreatic cancer is tough to treat. In 1997 the survival was 6 months. Now it's 12 months. So over 2.5 decades we've just about touching a year. So in such a tough cancer to treat, you essentially, any new development is welcome, and that's probably where this drug has its basis. These are the 3 major trials that we use commonly in clinical practice prior to NAL-IRI. It is FOLFIRINOX and Gemnab-paclitaxel versus gemcitabine, and maximum survival still date in a trial setting before NAL-IRI was around 11 months. And we either choose FOLFIRINOX or Gemnab-paclitaxel in clinical practice based on patient condition and PS. Gemcitabine, erlotinib, I think most of us have gone away from. So coming to Onivyde, 2016 you had the Napoli-1, which was in second line chemo, and Napoli-3 was in first line, and both of these are FDA as well as EMA approved. Coming to Napoli-3, which is the first line study, it's important to note that while FOLFIRINOX and Gemnab-paclitaxel are both options, I think even in India, and this is retrospective data from TMH, more patients get Gemnab-pacli as opposed to FOLFIRINOX based treatment for potential reasons of toxicity. But we don't have any firm evidence or guidelines to guide us which one to choose. This was one of the few retrospective real world evidence that was done, which showed that even though the trials had different numbers in terms of survival, a real world analysis said that largely the 2 regimens show similar outcomes in terms of overall survival. This is another one which is showing the same, that irrespective of what you use, overall survival as well as PFS are very similar between the 2 regimens. This is data from our institution where we did a match pair analysis between Gemnab-pacli and modified FOLFIRINOX. Again, the survival is 1 year. There's no difference, and this was around 350 patients. This is a meta-analysis showing the same. This was done for studies between 2015 and 18. There are no randomized head-to-head control studies, large phase 3 studies which have compared these 2 regimens, but what it does say is that if you look at the diamond, it's right at the center. So coming to Napoli-3, what they did was compare NAL-IRI-FOX with nab-paclitaxel and gemcitabine. And important thing to remember here is that advanced pancreatic cancers are tough to treat diseases, so doing phase 3 trials in these cancers is also tough. In terms of dosing, the thing to remember here is that oxaliplatin, the dose was 60 milligram per meter square. That's one thing that we need to know when compared to conventional FOLFIRINOX. The original protocol had used 85 milligram per meter square. This one has used 60 milligram per meter square. They compare it to standard dose Gemnab-pacli, which is an acceptable comparator in the current era. They didn't have any age limits, and as in most trials, they used ECOG PS 0 or 1. One of the points that was highlighted while this data was being published is that gem-nab-paclitaxel is a relevant comparator arm because it is one of the most commonly used treatment options world over, and a number of other trials have also used the same comparator. Now if you look at the survivals, the primary analysis of OS, it was 11 months with NAL-IRI-FOX and 9.2 months with gemcitabine-nab-paclitaxel. A sensitivity analysis showed that the difference in survival was slightly more with NAL-IRI-FOX, but essentially what you can see is that there's a difference in survival of approximately 2 months, and the curves after the first 2 to 3 months largely separate and kept separating. So this difference in survival was statistically significant. The p-value was 0.04. In terms of PFS, I mean, it is almost similar to the OS. The difference in PFS is around 2 months, 7.5 and 5.5 months with NAL-IRI-FOX. And in the management of pancreatic cancers, you're not going to get great survival differences. 2 to 3 months is largely what we get in most trials. This is just showing that what are the overall response rates in this study as compared to the gem-nab-pacli in the original MPAC study as well as the Prodigy trial. What you can see is that the objective response rate with this regimen is around 40%, and it's around 30% with FOLFIRINOX alone. These are all important secondary endpoints, but the fact is that the overall survival was obtained. These are just to guide you in terms of secondary endpoints. Disease control rates were around 60 to 70%, and that is normally what you see as well. In terms of adverse events, you would expect more adverse events with the combination of NAL-IRI-FOX. The grade 3-4 events for neutropenia were 14% only, which is probably lesser than what you would see with conventional modified FOLFIRINOX. With gem-nab-pacli, actually it was a little more, which is surprising. The other ones, vomiting, sensory neuropathy, were all manageable. The idea is that probably supportive care and things have improved, and that has led to this decrease in side effects. But if you look at the trial per se, slightly less than expected side effects were seen with the Onivyde containing combination. Coming to Napoli-1, treatment for a second line treatment in pancreatic cancers is essentially philosophy. If your patients live to 6 months, you're happy. So Napoli-1 was the trial that came first, where they actually had a 3-arm study of NAL-IRI alone, 5-FU alone, and the Napoli regimen. From a pancreatic cancer point of view, it was actually a large trial of more than 400 patients. So 400 patients in a second line pancreatic cancer trial is a large number. As I said, you're happy if your patients cross 6 months, and that's exactly what happened here. With the combination of NAL-IRI and 5-FU, the survival was 6 months, and with only 5-FU, it was 4 months. The PFS again was 3 months and 1.5 months. So, I mean, they set out to achieve an endpoint. They achieved the endpoint, but we do know that, these are very small numbers for patients as they are barely crossing 6 months. As expected, you would have greater side effects, and mostly they were diarrhea, vomiting, and nausea. There were patients who also had neutropenic sepsis, but again, largely manageable. Nothing extraordinary from this trial in terms of side effects. So this is what we think you can use in practice. If you're giving NAL-IRI-FOX as first line, then you shift to gemcitabine based therapy. If you're using gemcitabine based therapy as first line, you shift to 5-FU and Onivyde or FOLFOX. These are all options which give you 4 to 6 months of survival. Third line options are iffy. I don't think a majority of our patients actually reach third line therapy. So just to conclude, if you are using Onivyde as first line treatment, you can use it based on the results of the Napoli 3. It achieved its endpoint of improved overall survival. If you're giving it in second line, you have the basis of the Napoli 1 trial, where it also was slightly superior to 5-FU LV alone. With that, I'd like to thank you for your kind attention. Thank you so much.